Welcome back to the deep dive. Today we are diving into gain of function research, making super diseases to study them.
It is quite the topic to jump into today.
It really is. And I have to admit, when you first start reading about this stuff, it feels completely unreal.
Oh, absolutely. Like a movie.
Yeah, it feels like the plot of a science fiction thriller. You picture, like, scientists in underground bunkers engineering doomsday viruses.
Right, the classic evil genius trope.
Exactly. But the reality of what this research actually is and, why it happens every single day, it's so much more complicated.
And vastly more fascinating, I'd say.
Oh, without a doubt. It presents this profound dilemma to humanity. So for you listening right now, our mission today is to explore this hidden world.
We really want to pull back the curtain.
Yes, we're going to break down what it actually means to genetically alter a disease in a laboratory. And we'll do it without getting lost in a maze of, heavy medical jargon.
Which is very easy to do in this field, honestly.
Yeah, too easy. So we'll look at the powerful motivations driving scientists to take these risks. We're going to examine the hard mathematical odds of a catastrophic accident.
The numbers are, well, they're sobering.
They really are. And finally, we'll look at the political nightmare of trying to regulate something that is almost impossible to define. But before we get into the heavy science, I want to frame the core tension of this debate with a scenario.
Okay, lay it on me.
Imagine you are the chief of security for a massive, highly secure bank.
A bank with some very valuable assets.
The most valuable. And you just installed a brand new state-of-the-art vault. I mean, it's got the thickest steel, the most complex digital locks, the works.
Unbreakable.
Right. But to make absolutely sure that no thief can ever break in, you decide to hire a brilliant, world-renowned safecracker.
Okay.
And you give this safecracker a very specific job. You tell them, invent a brand new, never-before-seen tool that can shatter your own security system. Break into the new vault just so you can see exactly how they do it.
So you're intentionally asking someone to build a weapon against your own defenses.
Precisely. Now, the question that divides the entire world is this. Are you brilliantly preparing for the worst-case scenario? Like, ensuring you know exactly how to stop a future robbery before it even happens?
Oh.
Or did you just hand a master key to the universe, and now you have to pray every single night that this safecracker never loses that tool, never drops it in the parking lot, or, never accidentally leaves it where a real bank robber can find it?
That analogy, that perfectly captures the anxiety surrounding this whole field.
Right.
Because when researchers take a virus and intentionally alter it, to see what new dangerous things it's capable of, they genuinely believe they're building a necessary defense.
They see themselves as the good guys?
Exactly.
Yeah.
But the critics, they look at that exact same action and see the creation of an entirely new threat, a threat that just never needed to exist in the first place.
It's an incredibly heavy question, but, before we can even begin to debate the ethics or the safety of it, we have to look under the hood.
We need to define the basics.
Yeah. We have to understand what these scientists are actually doing when they, put on the gloves and sit down at the lab bench. What does it actually mean to tinker with a germ?
Well, at its most fundamental level, we can divide this genetic tinkering into two main categories.
Okay, two categories.
Yeah. The first category is what the scientific community calls loss of function.
Loss of function. Okay, I kind of like to think about this like a mechanic studying a mysterious, complex car engine.
That works perfectly, actually.
Ah,
Think of any germ, so a virus or a bacterium, as a tiny biological machine.
right.
And it has a very specific genetic toolbox that allows it to do its job, which is usually to infect a host and multiply.
Okay.
So loss of function is when scientists intentionally go in and, they take a tool out of that toolbox.
So going back to the mechanic analogy, if I want to figure out exactly what the spark plugs do in this engine, I can simply pull them out.
Yeah.
I remove that function entirely. And then when I turn the key and the engine fails to start, I've learned a vital piece of information about how the system operates. I weakened the car to understand it.
Right. You broke it to learn about it. If scientists remove a specific line of genetic code from a virus, and suddenly that virus can no longer attach to a human lung cell, they've just made a massive discovery.
Because they know that specific piece of code is the key to the infection.
Yes. By breaking the virus, they learn its vulnerabilities.
And taking tools away from a virus, that seems intuitively safe. You're making it less dangerous.
It generally is safe, yeah. But the flip side of that coin is the second category, and this is the one that generates all the controversy.
There we go.
This is gain of function. This is when scientists intentionally give a germ a brand new tool it never had before.
So if loss of function is pulling out the spark plugs, gain of function is taking that same car, strapping a military-grade rocket booster to the roof, and seeing how the aerodynamics change when you force it to go 300 miles per hour.
That is quite the visual, but yes, it's functionally accurate. You are pushing the system into an extreme unnatural state to see what it can do. You're giving the pathogen a new superpower.
Like what? What kind of superpower?
Well, maybe you alter a virus that normally only infects birds so that it can suddenly infect a mammal.
Hmm.
Or, maybe you take a virus that spreads through physical touch and you tweak its genetics until it can spread through the air in a sneeze.
Wow. And obviously, strapping a rocket to a car comes with infinitely more immediate danger than just pulling out a spark plug.
It absolutely does.
Hmm.
But one of the most surprising things about this entire field is that, well, this kind of biological manipulation is not some futuristic 21st century invention.
Really?
No, we have been doing this for a very long time. In fact, if you go all the way back to the 1800s, Louis Pasteur used loss of function to create the very first vaccines.
Wait, the 1800s? I always pictured this requiring modern, sterile laboratories with like multi-million-dollar microscopes.
Not at all. Pasteur was dealing with rabies, which is a terrifying and universally fatal disease.
Yeah, it's awful.
He didn't have gene editing machines, but he figured out that if he took the rabies virus and repeatedly passed it through the nervous systems of different animals, specifically rabbits in this case, and then dried out the spinal cords, the virus began to change.
Oh, I see. It was forced to adapt to a new environment, and the drying process weakened it.
Exactly. He created a version of rabies that had lost its function to cause severe fatal illness in humans.
Okay.
But, and this is the genius part, it still looked enough like rabies that the human immune system recognized it. It served as a wanted poster.
A wanted poster. I love that.
Yeah. The immune system studied the weakened virus, built up a defense, and then when the real deadly rabies virus showed up later, the body was ready.
So he was doing loss of function research over a century ago to create vaccines.
He was.
And that basic logic really hasn't changed, has it? We still use weakened viruses today to train our immune systems.
We rely on it heavily, actually. Think about the chickenpox vaccine. Most of us get it as children.
Yeah.
That vaccine is a live varicella zoster virus that has been intentionally weakened in a laboratory. It has lost the function of giving you full-blown chickenpox with the fever and the itchy blisters, but it retains the function of teaching your immune system how to fight off the real thing.
I was reading in our sources about a much more recent example, too, involving African swine fever.
Oh, yes, that's a great example.
Yeah, this is a devastating disease that can wipe out entire populations of pigs, which obviously has massive implications for global food supplies.
Huge economic impact.
Right. And scientists went into the genetics of that virus, identified specific genes, and intentionally deleted them. They literally stripped tools out of its toolbox to create a weakened version that successfully protected the pigs as a vaccine.
It's a textbook example of how vital this work is. Because without loss of function, we simply do not have some of our most critical medical defenses. It is the absolute cornerstone of modern vaccinology.
Okay, so that clearly explains what they're doing and why the safe version is so important. But I want to get into the nuts and bolts for you guys listening. How do you actually add or remove a tool from a microscopic virus?
Right. How does it actually work in practice?
Yeah, without getting into dense chemistry, how does the mechanic actually pull the spark plug or strap on the rocket?
Well, there are three primary methods that researchers use, and they range from surprisingly basic to incredibly high-tech.
Okay.
The first method is called serial passaging. And this is the old school approach. It's very similar to what Pasteur did with the rabbits. Think of it as a biological game of telephone.
A biological game of telephone. How does that play out in a laboratory?
Imagine you take a virus and introduce it to a dish full of cells. Or perhaps you infect a small laboratory animal.
Okay.
The virus enters the cells and starts multiplying. Now, viruses are incredibly sloppy when they copy themselves. They make constant tiny mistakes in their genetic code.
Like someone typing an essay really fast and making a bunch of typos.
Precisely. Those typos are mutations. Now, most of those typos make the new virus useless, and it just dies.
Right.
But every once in a while, a typo actually gives the virus a slight advantage in that specific environment. So the scientist takes the virus from that first dish and uses it to infect a second dish, then a third, then 100th.
You're passing the message down a long line of people, and the message changes slightly with every whisper.
And you are actively guiding how it changes. If you force the virus to keep jumping from one specific type of animal to another, you are putting it under immense survival pressure. It has to adapt to survive.
That makes sense.
Over time, through sheer trial and error and natural selection, those tiny typos accumulate into a massive genetic shift.
So you don't even necessarily need microscopic tweezers to go in and pluck out a specific piece of DNA. You just force the virus to run a specific obstacle course over and over until it evolves to be really good at running that course.
That's exactly it. That is serial passaging.
Yeah.
It relies entirely on the natural sloppy replication of the virus.
Okay, so what's the next method?
Now, the second method is much more modern and precise. It's called reverse genetics, which is basically building a virus from a blueprint.
Oh, well, how precise are we talking?
Extremely precise. Today, scientists can read the entire genetic sequence of a virus, every single letter of its code. They can load that sequence into a computer, look at it on a screen, and manually edit the letters.
Wait, really?
Yeah, they can delete a section or just type in a new one.
They treat the DNA like a Word document.
In a sense, yes. Once they have the edited blueprint on the computer, they use chemical synthesizers to physically manufacture that exact string of genetic material right there in the lab.
Wow.
And then they insert that man-made genetic material into a living cell. The cell reads the blueprint and just starts manufacturing the physical virus from scratch.
That is mind-blowing. The cell basically becomes an unwitting 3D printer for a custom-designed virus.
It's one of the most powerful tools in modern biology.
I can imagine.
And then we have the third method, which is reassortment, or mixing and matching. And this is actually something that happens in nature all the time.
Like how different strains of the flu combine to make a new flu season?
Exactly. If an animal, let's say a pig, gets infected with a bird flu virus and a human flu virus at the exact same time,
Bad day for the pig.
very bad day. But those two different viruses can infect the exact same cell in the pig's body. And inside that cell, as they are both multiplying, their genetic parts can get mixed up. They literally swap entire chunks of code.
So it's like taking the engine and heavy-duty suspension of a truck and combining it with the lightweight aerodynamic body of a sports car to see what kind of hybrid vehicle drives out of the garage.
That's a great way to picture it. And scientists can force this to happen in a highly controlled environment. They put two different viruses into the same dish and just let them swap parts to see what kind of hybrid emerges.
Okay.
So those are your three main avenues. The telephone game of serial passaging, the computer blueprint of reverse genetics, and the mixing and matching of reassortment.
Okay, I am entirely on board with using these methods for loss of function. I totally understand why we need to pull the spark plugs to build vaccines.
Right.
But this brings us to the most difficult part of the conversation. Why on earth would we ever use these tools to strap a rocket to a pathogen?
Oh, yeah.
I mean, why would we ever intentionally give a dangerous germ an entirely new capability that makes it more lethal or more contagious?
That is the ultimate question. The scientists who perform this work have a very robust rationale, and we can think of it as the wargaming defense.
The wargaming defense. Okay, let's dig into that. What exactly are they defending against?
They are defending against the inevitability of nature. Proponents of this research argue that we just cannot afford to sit around and wait for nature to throw the next devastating pandemic at us.
Because nature is already out there constantly mutating viruses in wild animals.
Exactly. If we just wait for those viruses to spill over into humans naturally, we are perpetually playing catch-up.
So they want to figure out what a virus is capable of doing before it actually manages to do it in the real world.
Yes. They want to be proactive. They want to build the antidote before the poison even finishes naturally evolving.
You know, this reminds me heavily of the cybersecurity world.
How so?
Well, large companies and governments regularly hire what are known as white hat hackers.
Oh, right.
These are brilliant computer experts who are paid to break into a company's network. Not to steal money, but to expose the vulnerabilities. And to do that effectively, a white hat hacker sometimes has to write custom malicious software. They have to build a digital weapon just to see if the company's firewall can stop it.
That's a great point.
You have to create the threat to understand how to defeat the threat.
That is an excellent way to understand their mindset. These researchers view themselves as biological white hat hackers. They are actively probing the defenses of human public health.
Let's ground this in a real-world example from the sources, because this is where the theory hits reality in a very, very intense way. Back around 2011, two prominent scientists, Ron Fauché in the Netherlands and Yoshihiro Kawaka in the US and Japan, they conducted a series of experiments that sent shockwaves through the scientific community. They were working with the H5N1 bird flu.
Yes, H5N1 is a fascinating and frankly terrifying virus.
Yeah.
It is incredibly lethal to birds.
Yeah.
And on the rare occasions when a human catches it directly from handling a sick bird, it has an astronomically high fatality rate in humans.
Yeah.
But historically, it had a major biological limitation.
Right, it couldn't spread from human to human easily.
Correct. And the reason for that is temperature. A bird's respiratory tract is significantly warmer than a mammalian respiratory tract.
Okay, I didn't know that.
Yeah, so the bird flu virus had evolved to thrive in that warm environment. When it gets into the cooler temperature of a human nose and throat, it struggles to survive and replicate efficiently.
So even if one person caught it from a chicken, they weren't going to sneeze on an airplane and infect 100 other people. The biological barrier protected us.
Exactly. But Fauché and Kawagoka wanted to know a very specific, frightening piece of information. They wanted to know, was that biological barrier permanent?
Oh, boy.
Right. Could this deadly bird virus figure out a way to adapt to the colder mammalian respiratory system and spread through the air?
Because if a virus with the lethality of bird flu suddenly gained the transmission speed of the common cold, I mean, that is a civilization-altering event.
It is the ultimate nightmare scenario. So, to find out if it was biologically possible, they set up an experiment using the serial passaging method, the game of telephone. But instead of Petri dishes, they used ferrets.
I always wondered why ferrets pop up so often in these studies.
It's because ferrets are the gold standard for studying respiratory viruses like the flu. Their lungs and respiratory tracts are remarkably similar to humans. They experience the flu very much like we do. And crucially, they sneeze.
Okay, so picture the laboratory setup for us. How do they actually run this obstacle course?
They placed a ferret infected with the bird flu in a cage. Right next to it, separated by a perforated barrier, they placed a healthy ferret.
Okay.
They couldn't touch each other, they couldn't share food or water, but they breathed the exact same air.
They were testing for airborne transmission?
Initially, nothing happened. The virus couldn't cross the air gap. The biological barrier held.
Well, that's good news.
But the scientists didn't stop there. They took swabs from the sick ferret's nose, transferred that virus directly into another ferret's respiratory tract, let it multiply, took a swab from that one, and put it into a third.
So they were forcing the virus to repeatedly replicate inside the cooler environment of the ferret's nose. They were just turning up the evolutionary pressure.
Precisely. They were actively selecting for any tiny typo, any mutation, that allowed the virus to survive just a little bit better in a mammal. And after roughly 10 passes through different ferrets, something chilling happened.
The virus mutated.
It gained a new function.
The rocket booster.
Yes. They put an infected ferret next to a healthy one, separated by the barrier, and the healthy ferret got sick. The bird flu had successfully altered its genetic code to spread through respiratory droplets in the air between mammals.
Oh, wow. So they proved that the nightmare scenario was biologically possible. The barrier was not permanent at all.
They did. And there's a quote from Ron Fauché in the sources that perfectly encapsulates the dual nature of this work. Reflecting on the results, he said he was, quote, intrigued in how little things could kill large animals and humans.
That is a very clinical, almost chilling way to look at something that could cause a global catastrophe.
It is.
But I understand the raw scientific curiosity. He wanted to dismantle the engine to see exactly how the pistons fired. He wanted to know the enemy.
And the proponents of the research argue that this knowledge is invaluable. Because once Fauché and Kawaoka identified the exact genetic mutations that allowed the virus to go airborne in the lab, scientists across the globe suddenly knew what to look for.
Oh, they had the wanted poster.
Exactly. They had a wanted poster. They could go out and monitor wild bird populations, sequence the viruses they found in nature, and if they ever saw those specific mutations starting to develop naturally, they could sound the alarm immediately.
Okay, I see the logic. It is an early warning system. We map the genetic pathway in a secure vault so we can spot it out in the wild.
That is the primary argument. But the wargaming defense goes beyond just surveillance. Think about how we test our medicines. How do we know if the antiviral pills we rely on today will still work against the viruses of tomorrow?
Well, viruses evolved to beat drugs, just like bacteria evolved to beat antibiotics.
Exactly. Pathogens are brilliant escape artists. So researchers will put a virus in a dish, flood it with a common antiviral medicine, and wait. Most of the virus dies.
But not all of it.
Right. Inevitably, a tiny fraction survives because they happen to have a random genetic typo that gives them resistance to the drug.
They gain the function of drug resistance.
We call them escape mutants. By intentionally forcing the virus to gain this function in the lab, scientists can study the mechanics of how it escaped. If they understand the escape route, they could design a better lock.
Makes sense.
The sources detail a perfect example of this. Researchers studied how influenza viruses rely on a very specific viral protein to spread from cell to cell. By forcing the virus into a corner, they understood the protein's structure better, which directly led to the creation of a highly effective antiviral drug called peramivir.
So forcing the mutation directly led to a better
It
medicine.
did. And there's one more critical piece to the proponent's argument, and it is known as the surrogate animal problem.
This one fascinated me. Walk me through the surrogate problem.
Let's imagine a brilliant scientist working in a lab discovers a chemical compound that perfectly destroys a specific human virus in a petri dish. It's a potential cure. But the rules of medical science dictate that you cannot simply take a chemical from a glass dish and inject it directly into a human patient. You have to prove it's safe and effective in a complex living organism first.
Right, you have to do animal trials, usually starting with mice.
But here is the massive roadblock. What if the human virus you're trying to cure does not naturally infect mice? A mouse's biology is different. The virus just bounces off their cells.
Oh, if the mouse can't catch the disease, you can't test the cure on the mouse.
Exactly. You're completely stuck. So what is the solution? The scientist has to intentionally genetically alter the human virus. They have to use gain-of-function techniques to give the human virus a new superpower, the ability to infect and sicken a mouse.
So they have to make the pathogen more versatile and adaptable just to test the medicine they made for it.
It's a paradox, but it's true. The sources highlight an example involving a dangerous bacterium called Pseudomonas aeruginosa.
Hard to say.
Very. But researchers had to fundamentally alter how this bacterium behaved so it would successfully infect a whole living system, a mouse, allowing them to study how it causes disease and how to stop it. Without gaining that function, the research hits a brick wall.
Okay, so we have early warning surveillance, we have testing antiviral escape routes, and we have the surrogate animal problem. But there's one more argument that proponents make, and it completely shifts the perspective.
Yes, the bioreactor argument.
Right. They argue that while we sit here agonizing over whether it is safe to do this in a secure vault, nature is out there acting as a giant, unregulated bioreactor.
This is a crucial point. Nature is running these exact same experiments every single second of every single day. Trillions of viruses are multiplying in billions of animals, birds, and insects globally. They're constantly mutating, swapping parts, and crossing species barriers.
Nature doesn't need to fill out a risk assessment form. And the story from the sources about the Dutch seals perfectly illustrates this.
It's a stunning historical coincidence. We talked about Ron Fouchier's ferret experiments, where he made the bird flu airborne.
Yeah.
Well, when those results were announced, there was a massive public outcry. People were terrified. So the government stepped in and temporarily shut down his lab to assess the danger. They put a hard pause on the research.
So the scientists are locked out of the vault. The supervirus is frozen.
Right. But while the politicians and scientists were arguing about safety protocols, something was happening in the wild. Off the coast of the Netherlands, wild seals began dying from a respiratory illness.
Just spontaneously.
Yes. When scientists sequenced the virus killing the seals, they discovered it was a form of bird flu. And incredibly, nature had arrived at the exact same airborne mutations that Fouchier had artificially generated in his lab.
Wow. So while humans were arguing about whether the experiment was too dangerous to conduct in a vacuum-sealed room, nature just ran the experiment in the open ocean.
Exactly. The proponents point to that event and say, look, nature does not take days off. The mutations are going to happen. The pathogens are going to evolve. We can either bury our heads in the sand, or we can study the mechanics in a controlled environment so we're ready when nature eventually hits upon the winning combination.
I hear that argument, and the story of the seals is compelling. But this is exactly where I have to step in and pivot to the other side of the debate.
Fair enough.
Because I am looking at the reality of this, and a wild seal swimming in the frigid waters of the North Sea is profoundly different from a highly concentrated, perfectly engineered vial of supervirus sitting in a laboratory.
The environment is entirely different.
Exactly. That laboratory is likely situated in the middle of a major city, surrounded by millions of people, connected by international airports. The proponents talk about perfect control. But what happens if our brilliant white hat hacker accidentally drops the master key in the bank lobby? What if they accidentally hit send on the malicious code and wipe out the entire financial system?
You are outlining the exact foundation of the critics' case. And their primary argument boils down to one undeniable reality: human error.
Because at the end of the day, it doesn't matter how thick the steel vault is or how sophisticated the negative pressure air filtration system is. The people working inside those vaults are human beings.
And human beings, no matter how highly trained, no matter how many PhDs they hold, make mistakes. It is an inescapable law of statistics.
Right.
If you run enough complex procedures over a long enough timeline, someone is going to slip. Someone is going to mislabel a vial. Someone is going to have a bad day and forget a protocol.
And we don't have to speculate about this. The sources provide a terrifying list of historical proof where some of the most prestigious, highly secure laboratories in the world made catastrophic blunders.
The incidents are sobering. We are talking about facilities like the Centers for Disease Control and Prevention, the absolute pinnacle of biosecurity.
The CDC.
In one deeply unsettling instance, a National Health Agency laboratory thought they had properly deactivated a batch of anthrax.
Deactivated meaning they killed the bacteria so it was safe to handle.
Right. Believing it was harmless, they shipped these samples out to other laboratories that did not have the high-level security containment required for live anthrax.
Oh, no.
The scientists in those receiving labs worked with the samples using only basic safety gear.
Because they trusted the label.
But the label was wrong. The protocol to kill the anthrax had failed, and nobody double-checked properly. They had unknowingly shipped live, lethal anthrax through the mail to under-equipped labs.
That is terrifying. Just a simple procedural error leading to massive exposure, and then there's a smallpox story.
That one sounds entirely made up, but it is fully documented. Smallpox is arguably one of the most devastating diseases in human history.
Yeah, terrible.
We eradicated it globally through a massive vaccination campaign, and the remaining live samples are supposed to be locked away in only 2 ultra-secure fortresses in the entire world.
Fort Knox for biological weapons.
Exactly. But a few years ago, scientists were cleaning out an old, forgotten storage room on a government campus. And sitting there, in an ordinary cardboard box, they found forgotten glass vials containing freeze-dried smallpox virus.
A disease that killed hundreds of millions of people just sitting in a cardboard box in a closet because someone decades ago forgot to log it properly.
It perfectly illustrates the critic's point. Biosecurity relies entirely on human memory and human adherence to the rules.
It's so fragile.
In another incident highlighted in the sources, researchers working with bird flu accidentally cross-contaminated a relatively harmless strain with a highly pathogenic, deadly strain.
They mixed them up on the bench.
Yes, and then, thinking they only had the harmless strain, they shipped it out to an unsuspecting laboratory. The scientists receiving it thought they were handling a common cold, essentially, when in reality they were holding a potential pandemic pathogen.
There is a very clear pattern here. The high-tech containment suits and the vault doors don't fail. It's the moment a human being makes an assumption. They assume the virus is dead, or they assume the vial is labeled correctly, so they take it out of the secure zone. The technology is perfect, but the human interface is fundamentally flawed.
And the consequences of those human flaws can be global. There is a very prominent, highly credible theory among virologists that a laboratory escape has actually sparked a global pandemic in modern history.
You're talking about the 1977 flu pandemic. I brought this up in my notes because the detective work behind it is incredible.
The Russian flu, as it was called. In 1977, a wave of H1N1 influenza swept across the globe. But epidemiologists noticed something very strange about how it was behaving.
It was almost exclusively infecting young people, right? People in their 20s and younger.
Yes. Usually a new flu strain hits everyone. The fact that older people were mostly unaffected meant that their immune systems had seen this exact virus before. They had pre-existing immunity.
Which implies the virus had circulated in the past.
So Virologists started looking at the genetic code of this 1977 virus, and they were stunned. They compared it to a frozen strain of H1N1 that had circulated widely back in 1950.
27 years earlier.
Viruses in the wild are constantly mutating. They have a known evolutionary clock. A virus from 1977 should look significantly different from a 1950 virus because of 27 years of constant typos and adaptations.
But it didn't.
It didn't. The genetic code of the 1977 virus was virtually identical to the 1950 strain. It was missing 27 years of evolution.
As if it had been perfectly preserved in a time capsule or a laboratory freezer.
The overwhelming consensus among experts is that the 1977 pandemic was a 1950 strain of flu that was being studied in a laboratory, accidentally infected a researcher, and walked right out the front door to sweep the globe.
Wow. So we have a historical precedent for exactly what the critics fear. But, anecdotes and historical mysteries are one thing. The actual mathematical odds of a disaster.
This is where we look at the work of Mark Lipsitch and Alison Galvani. They are epidemiologists who wanted to strip away the emotion and look purely at the statistical probability of a laboratory-acquired infection leading to a global outbreak.
How do you even calculate the math on something like that?
You start by establishing a baseline rate of failure. They look at historical data regarding high-security biological laboratories. They gathered records of every time a worker accidentally poked themselves with a needle, or a centrifuge leaked, or someone got sick. Based on decades of data from biosafety level 3 and 4 labs, they calculated a conservative accident rate.
And what was that baseline rate?
Roughly 2 laboratory-acquired infections for every 1,000 years of laboratory operation.
2 infections per 1,000 lab years. I mean, that sounds incredibly low. That sounds like a phenomenal safety record.
It is a very good safety record.
Yeah.
But when you are dealing with engineered pandemic pathogens, even a tiny failure rate becomes terrifying when you extrapolate it out. Lipsitch and Galvani ran a hypothetical, highly conservative scenario.
Well, let's walk through their scenario.
They said, let's imagine a world with strict regulations. We only allow 10 specific laboratories globally to conduct this kind of advanced gain-of-function research on transmissible viruses. Just 10 labs. And we only allow them to run these programs for a period of 10 years.
So 10 labs operating for 10 years, that's 100 lab years of total exposure.
Exactly. When they applied the historical baseline failure rate to that small controlled scenario, the math produced a chilling result. The probability that at least one of those 10 labs would experience an infection that escaped the facility during that decade was over 18%.
18%, almost a 1 in 5 chance of a catastrophic breach, just from 10 labs over 10 years.
And remember the nature of what they are studying. If a researcher gets infected with a naturally occurring, hard-to-transmit virus, it might end with them. But if they get infected with a virus specifically engineered in that lab to spread rapidly through the air via human sneezes, it only takes one person stopping for coffee on their way home to initiate an uncontainable global chain reaction.
1 in 5 odds of sparking a pandemic is an intolerable level of risk. But I want to push the critics' argument even further. Because the math is terrifying, yes, but the critics also attack the foundational premise of the research itself. They question whether the wargaming predictive maps even work.
Yes, this is a profound scientific critique, and it centers around a very complex biological concept mentioned in the sources called epistasis.
Epistasis. Okay, I spent a lot of time trying to wrap my head around this, and I want to try translating it into an analogy to make sure I understand it correctly. Tell me if this holds up.
Go for it. Let's hear it.
Let's say I want to build an incredibly fast vehicle. I go into a high-end garage, I take the steering wheel out of a precision-engineered Ferrari, and I carry it over to a rusty farm tractor. I unbolt the tractor's steering wheel and I bolt the Ferrari steering wheel in its place.
Okay, I am following you.
Just because I added a component from a 200-mile-per-hour sports car, that farm tractor is not going to suddenly start going 200 miles per hour down the highway.
No, definitely not.
The background matters. The heavy engine, the massive treaded tires, the terrible aerodynamics, the whole rest of the tractor's structure prevents that high-performance steering wheel from doing what it was designed to do.
That is an absolutely brilliant analogy for epistasis. In biology, a genetic mutation does not exist in a vacuum. Its effect is highly intimately dependent on the specific genetic background, the rest of the virus that it is attached to.
So how does that tractor analogy tear down the wargaming defense?
Let's look back at the ferret experiments with the bird flu. The scientists used a very specific, carefully cultivated, pampered laboratory strain of the H5N1 virus. They forced it to mutate, and it gained the function of airborne transmission.
Right.
The proponents cheer and say, "Excellent, we have our predictive map. If we ever see this exact mutation in a wild bird, we know it's about to go airborne."
But the wild bird virus is the tractor.
Exactly. The bird flu circulating in a wild duck in a muddy pond in Asia is biologically chaotic. It has a wildly different genetic background than the pristine lab strain. The critics argue that if that wild virus happens to naturally develop that exact same Ferrari mutation you identified in the lab, it might not go airborne at all.
Because of the background.
Yes, the messy tractor background of the wild virus might suppress the mutation entirely. It might just fizzle out and do nothing.
Wow, so the entire premise, the idea that we're building these perfect predictive maps to monitor global outbreaks, might be fundamentally flawed. The maps might be pointing to destinations that don't exist in the real world because the real world is too messy.
That is the core scientific pushback. The critics say we are accepting an 18% mathematical risk of a global catastrophe to generate predictive maps that might be practically useless for actual public health surveillance.
And if we look at our most recent history, our assumptions about what constitutes a dangerous virus have been completely upended anyway.
You are referring to the harsh lesson the world learned from COVID-19.
Right. Before COVID, my understanding of pandemic wargaming was heavily influenced by Hollywood. To be a true civilization-threatening supervirus, a pathogen had to be highly contagious, spreading like wildfire, and highly lethal, killing 50 or 60% of the people it infected. We were looking for airborne Ebola.
That was the old paradigm of a potential pandemic pathogen. Extreme transmissibility combined with extreme virulence. If a virus didn't kill a massive percentage of its hosts, it was generally considered manageable.
But COVID-19 proved that entire philosophy wrong. Here we had a virus with an infection fatality rate that, for much of the healthy population, was incredibly low fractions of a percent. By the old definitions, it wasn't an apocalyptic killer.
But its superpower was its stealth and its speed. Because it spread so incredibly fast and often asymptomatically, the sheer math overwhelmed the low fatality rate.
Exactly. Even if a virus only kills 0.5% of the people it infects, if it infects billions of people in a matter of months, the absolute number of deaths is staggering. It caused massive global economic collapse, shut down societies for years, and pushed healthcare systems to the brink of total failure.
It proved that you do not need to engineer a 60% death rate to bring the modern world to its knees. If a virus gains the function of rapid, silent spread, the fatality rate becomes secondary. The volume of infections alone will shatter society.
So, summarizing the critic's position, the risk of human error is guaranteed. The mathematical odds of a lab leak are deeply concerning. The predictive maps generated by the research might be useless because of the epistasis tractor problem. And our fundamental definitions of what makes a virus dangerous are outdated anyway.
That is a very robust summary of why so many scientists are vehemently opposed to intentionally creating superbugs.
Okay, listening to all of this, the math and the risks, a very practical question comes to mind. We talked about 10 theoretical labs in the risk model, but what is the actual reality? How massive is this landscape? Are we dealing with a few isolated academics, or is this a massive industry?
To understand the true scale, we have to turn to the data. And the sources provide a comprehensive, eye-opening report from CSET, the Center for Security and Emerging Technology. They decided to map the entire global footprint of this kind of research.
I brought the numbers from the CSET report, and they genuinely stunned me. They analyzed published scientific literature from the year 2000 all the way up until mid-2022. And they found over 7,000 published papers that fit the criteria for gain or loss of function research.
7,000 publications. That is a mountain of data. It completely shatters the illusion that this is a fringe or niche field of study. It is a massive, highly integrated, global scientific endeavor.
And when you look at who is doing it, the United States is the undisputed heavyweight. U.S.-affiliated researchers were involved in roughly 53% of all those publications.
Over half the global output has U.S. ties, and Chinese researchers were involved in about 21%. Beyond that, you have massive contributions from the UK, Germany, Japan, France, and many others. It is a worldwide network of laboratories.
But what exactly are thousands of scientists tinkering with? Are there 7,000 papers on how to make airborne bird flu?
Thankfully, no. The scope of the research is incredibly broad. The majority of it, about 64%, focuses on viruses. This includes studying seasonal influenza, herpes viruses, and coronaviruses like SARS and MERS to understand how they infect cells.
That makes sense. But the other chunk of the pie surprised me. The report notes that almost 29% of this research is actually focused on bacteria. Why are scientists genetically altering bacteria?
Primarily to fight one of the greatest slow-moving crises in modern medicine, antibiotic resistance.
The terrifying reality that our drugs are stopping working.
Exactly. Bacteria are constantly evolving to survive the antibiotics we use to kill them. If we lose effective antibiotics, modern medicine effectively collapses. Simple surgeries become lethal risks. Minor scrapes can lead to fatal infections.
So scientists use these techniques to fight back.
Yes, they use loss of function to find the bacteria's weak points.
Yeah.
And they use gain of function to force the bacteria to develop resistance in the lab so the scientists can study the mechanism and invent new, stronger antibiotics before the old ones fail completely.
It highlights the undeniable public health value embedded in this work. The report also found that about a quarter, 24% of all this research is directly related to developing vaccines.
And this brings us to the central frustrating reality of the CSET data. You cannot look at these 7,000 papers and sort them into two neat piles labeled safe, good vaccine research and dangerous, bad supervirus research. The data shows an incredibly intertwined reality.
The report says almost half the study's 46% are strictly loss of function. Just pulling the spark plugs, that seems clear-cut.
But look at the next statistic. 29% of the studies resulted in both gaining a function and losing a function in the exact same experiment.
Because biology isn't a neat engineering project.
Exactly. Let's say a scientist is trying to manufacture a massive amount of a virus to produce a vaccine. They want the virus to replicate much faster in the factory vat. That is a gain of function, gaining growth speed. But in the process of forcing the virus to adapt to growing in a stainless steel vat, the virus might naturally lose its ability to effectively infect a human lung.
Yeah.
That is a loss of function. They happen simultaneously. You cannot surgically separate the outcomes.
Okay, the entangled nature of the results makes it complicated. But here is the piece of data from the report that I found most alarming. It is the technology they are using. I assumed that to engineer a pathogen, you need a high-tech, billion-dollar CRISPR gene editing suite.
That is the most dangerous misconception in this entire debate. The CSET data reveals a massive low-tech method problem. The barrier to entry for altering a pathogen is shockingly low.
Remember serial passaging, the biological game of telephone we discussed?
The report found that 21% of all the research analyzed uses that incredibly basic method. But here's the truly stunning statistic. If you look only at the studies that resulted exclusively in a gain of function, the studies where scientists purely added a new capability to a germ, 42% of them used this low-tech serial passaging method.
Wait, really? Nearly half of the pure gain-of-function work doesn't require futuristic DNA printers.
No, not at all.
I'm going to use another analogy here because this is vital to understand. Let's say I want to radically alter the genetics and the physical structure of an animal. Let's say a wolf.
Okay, a wolf.
If I want to turn a wolf into a pug, I don't need a state-of-the-art genetics laboratory. I don't need microscopic tweezers to splice wolf DNA. All I need is time, several generations of wolves, and the basic concept of selective breeding.
Right.
I just take the smallest wolves, breed them together, take their smallest offspring, breed them, and wait. Nature does the genetic engineering for me.
And serial passaging is exactly the same mechanism, just applied to viruses on a microscopic, highly accelerated timeline. You are selectively breeding the pathogen by controlling its environment.
Which means that anyone with basic foundational laboratory equipment, some cell cultures, a controlled environment, and some patients can theoretically perform this work. You don't need enriched uranium and giant centrifuges like you do for a nuclear weapons program. The tools are everywhere.
The democratization of the tools makes policing this field a logistical nightmare.
And that leads us perfectly into the final massive question. If the math tells us the risk of catastrophe is real, and the data tells us that thousands of scientists are doing this globally with easily accessible tools, why can't the government just write a rule to fix it?
If only it were that simple.
Why can't a president or a prime minister simply slam a gavel and say, keep doing the good vaccine work and immediately stop creating superviruses? Problem solved.
If only it were a matter of political willpower. The reality is that the U.S. government, spanning multiple administrations, has been locked in a bitter political and scientific tug-of-war over this for more than a decade. They have tried to write the rules, and they continually run into walls.
And I want to be exceptionally clear here for the listener. We are not taking political sides in this. Both left-leaning and right-leaning administrations have wrestled with this beast and come away bruised. We are simply going to look at the history of how they tried to manage it, because it perfectly illustrates why the task is nearly impossible.
The modern battle really began in 2014, during the Obama administration.
This was right after that terrifying cluster of lab accidents we talked about. The CDC shipping live anthrax, the smallpox found in the cardboard box.
Yes, the public was highly alarmed, and the scientific community was deeply divided over the ferret bird flu experiments. Facing intense pressure, the government took a drastic step. They issued a mandatory pause. They halted all federal funding for any gain-of-function research involving influenza, SARS, and MERS viruses.
They basically froze the bank accounts to force the laboratories to stop working while they figured out a plan.
And that pause triggered a literal civil war within the scientific community. Two highly vocal opposing factions emerged.
On one side, you had the critics, who organized under the name the Cambridge Working Group. They issued public letters arguing that engineering potential pandemic pathogens in a lab was an existential risk to humanity, and they demanded the pause be made permanent.
And on the exact opposite side, you had a faction calling themselves Scientists for Science. They argued with equal passion that the funding pause was disastrous. They believed it was crippling our ability to develop vaccines and understand emerging threats, leaving the world vulnerable to the next natural pandemic.
So you have the brightest minds in virology completely deadlocked. What does a government do with that?
They spent 3 years studying it, convening panels, and trying to find a compromise. Finally, in 2017, the government lifted the pause. They replaced it with a new framework that relied on risk-benefit assessments.
Basically, a bureaucratic committee. Before you get the funding, you have to prove to a panel that the potential benefit of your experiment to society outweighs the potential risk of causing an outbreak.
Which sounds incredibly rational on paper. But the controversy didn't disappear. Over the last several years, both the Trump and Biden administrations have faced intense, renewed pressure to crack down. There have been executive orders, congressional hearings, and constant threats to completely pull federal life sciences funding from any institution that crosses the line into dangerous research.
So the politicians are trying to use the power of the purse. If you build a super virus, you lose all your grant money. Why doesn't that threat solve the problem?
Because when you take that political threat and try to apply it to a real-world laboratory, you hit 4 massive, insurmountable problems. The first is the definition problem.
Defining what is actually dangerous.
Exactly. The term gain of function is actually hated by most microbiologists because it is so broad it becomes meaningless. Yoshihiro Kawaoka, the scientist from the ferret studies, points out a massive legal flaw. Let's say a researcher creates a brand new, highly contagious virus that has never been seen in nature, that is obviously high-risk gain of function. But what if another researcher takes a human virus and alters it slightly so that it only infects mice, specifically so they can test a life-saving cure? And the altered virus is completely harmless to humans. Technically, under the broad definition, that is also gain of function. But they present entirely different levels of threat. Where exactly does the government draw the legal line between the two?
If the law is vague, scientists don't know if they are going to lose their careers just for testing a cure.
Which leads directly to the second massive hurdle, the accidental gain.
This implies scientists don't actually know what the outcome will be before they run the experiment.
Which is the very definition of an experiment. Biology is deeply unpredictable. Let's say a scientist is doing a completely safe standard loss-of-function study. They intentionally delete a gene from a virus to weaken it for a vaccine. That is their stated intent.
Pulling a spark plug.
But the organism compensates. Because that gene was deleted, the virus genetically reroutes itself. And suddenly, unintentionally, it starts multiplying twice as fast as it did before.
They intended a loss of function, but they accidentally triggered a gain of function.
Exactly. How can a government write a law regulating an accident before it happens? You cannot penalize a scientist for an unpredictable biological reaction. The intent was safe, the outcome was dangerous, and the law has no way to handle that nuance.
That is a fascinating paradox. You can't legislate the unknown. What is the third problem?
The shared toolbox. We established that the methods used for dangerous gain of function, like serial passaging and reverse genetics, are the exact same methods used for safe, vital loss-of-function vaccine research.
The mechanic uses the same wrench to pull the spark plug as he does to bolt on the rocket.
So if a government decides that the tools themselves are too dangerous and bans them,
You throw the baby out with the bathwater. You instantly ban the ability to create vaccines. You paralyze the entire field of virology because you confiscated their wrenches.
precisely. And that brings us to the fourth and perhaps most surprising problem: broader impacts.
Because these tools aren't just used to study human diseases.
Not at all. The techniques of adding or removing genetic functions are foundational to almost every biological science today. If a politician writes an overly broad law banning these techniques to stop superviruses, they could accidentally devastate completely unrelated fields.
Like what?
Agriculture, for example. Scientists routinely use these exact genetic techniques to create better, more resilient crops, or to design bacteria that fix nitrogen in the soil, which reduces the need for extensive chemical fertilizers.
Oh, wow. What about environmental cleanup? I know they engineer bacteria for that.
Exactly. In
Oh.
environmental science, researchers are using gain-of-function techniques to literally alter bacteria so they can digest toxic chemicals. They take a germ and give it the function of eating the toxic oil sludge at a polluted Superfund site.
That is incredible. So, a clumsily written law trying to prevent the next flu pandemic could accidentally make it illegal for scientists to clean up a toxic waste dump or engineer cheaper, hardier food supplies for a growing population.
Yes, the biological sciences are a deeply tangled web. When regulators reach in and try to forcefully pull out the dangerous threads, they risk unraveling the entire tapestry of modern scientific progress.
Okay, let's step back and look at the massive picture we just painted. We have scientists conducting research that could theoretically map out the next global pandemic or provide the crucial mechanism to defeat antibiotic resistance.
Right.
But to achieve that, they must run experiments that carry a mathematically terrifying probability of accidental escape. The tools they rely on are so fundamental and low-tech that a comprehensive ban is likely unenforceable. And even if you tried to enforce it, you would accidentally outlaw vaccine development, agricultural innovation, and environmental cleanup.
It is the ultimate dual-use dilemma of our time. The very scientific processes that hold the potential to save millions of lives also hold the potential to destroy them.
It leaves your head spinning. As we wrap up this incredibly complex deep dive, I want to leave you with one final, lingering thought to mull over. Because throughout this entire conversation, we have focused heavily on human actions. The government regulators, the vault doors, the scientists in the lab coats.
Yes, but we cannot forget the ultimate wild card in all of this.
We know for a fact that nature is performing these biological experiments every single second in billions of animals worldwide. The seals in the ocean do not need to apply for a research grant to mutate a deadly virus.
So,
And we also know from the CSET data that creating an enhanced virus does not require futuristic technology, just patience, test tubes, and the basic selective breeding of serial passaging.
the biological game of telephone.
Exactly. So here's the provocative question to take with you. If a government decides the mathematical risk is simply too high and they pass a law banning all federal funding for this research, shutting down the highly visible, transparent, heavily regulated laboratories, does it actually eliminate the danger?
Or does it merely force the research out of the light and into the shadows?
Right. Does a ban actually protect us from a catastrophe, or does it just guarantee that we are entirely blind to the viruses that nature or perhaps someone else in an unregulated lab halfway across the world is brewing right now?
It is a profound, unsettling question. Do we want the safecracker working inside the bank where we can monitor every move he makes? Or do we fire him, ban his tools, and just hope he doesn't take his skills to the shadows?
Thanks for coming along on this deep dive with us. Remember to follow us on your favorite podcast platform, and see you on the next dive.